In this Explain Everything, we're going to be looking at water and lipid-soluble hormones. So let's just review the terminology. Water-soluble hormones are going to be hydrophilic. Lipid-soluble hormones are going to be hydrophobic. Water-soluble are going to be lipophobic, and lipid-soluble will be lipophilic. Looking at their capability of crossing the phospholipid bilayer, let's just quickly look at the bilayer, remembering that these are phospholipids with their tails in the interior of the membrane being hydrophobic. The concept that you have to remember is that like dissolves like, which means hydrophilic will dissolve hydrophilic, hydrophobic will dissolve hydrophobic, which means that in order to pass through the hydrophobic interior of this membrane, the molecule must be hydrophobic, which means that lipid-soluble molecules are going to be able to pass through the phospholipid bilayer and go from the extracellular fluid into the intracellular fluid. It also means that it's going to be able to pass the two phospholipid bilayers that make up the nuclear envelope and actually go into the nucleus. Let's first look at the lipid-soluble hormones. Because they are lipid-soluble, we've discussed previously, they are going to be hydrophobic, which means they are not going to be able to be transported, or they're not going to be soluble in the aqueous environment of the blood. So they are going to need the aid of a carrier protein. So they'll be transported in the blood via this carrier protein. When they meet their target organ, their target tissue, they will leave the plasma and then enter the cell, remembering that they can freely cross the plasma membrane and go into the cell. Okay, once in the cell, they are then in the aqueous environment of the cytoplasm, okay, and they are then going to either have to bind to a receptor in the cytoplasm, or they can bind to a receptor within the nucleus. Okay, so once again, freely passing through the phospholipid bilayer and passing through the two phospholipid bilayers that makes up the nuclear envelope. Once inside the nucleus, these hormones are then going to alter gene expression, okay, and altering transcription, ultimately altering the synthesis of a protein. The alteration of this protein synthesis then alters the physiology of the cell, and that is the mechanism of action of these steroid hormones, for example, or the thyroid hormones or nitric oxide. Thyroid hormones are lipid-soluble. Let's just review the control of their release. They, at the level of the hypothalamus, it's releasing thyroid-releasing hormone, going to the anterior pituitary, stimulating the release of thyroid-stimulating hormone, which is then stimulating these thyroid follicles to release the two hormones T3 and T4. Just reviewing for a minute, what happens is in the thyroid follicle, the two hormones are stored in an inactive gel-like substance called TGB, okay, which stands for thyroglobulin. When T3 and T4 are released into the blood, remembering that these are going to be hydrophobic, they're not going to be soluble in the blood, so they are bound to carrier proteins, and the name of this carrier protein is thyroid binding globulin. Okay, now the problem being that these two terms are quite similar, so you have to remember which way to, or how to figure those out. Okay, so the TGB versus the TBG. Now taking a look at water-soluble hormones, they're going to have no problem in the blood because they are hydrophilic, so they will not need a transport protein as they move through the blood. The problem with these, however, is when they get to their target tissue, they are not going to be able to diffuse through the plasma membrane. Therefore, they need receptors. So if we take a look at the cell, it is going to have receptors embedded into that phospholipid bilayer, and these hormones are then going to bind to the extracellular receptors. However, that message must get to the inside of the cell, and this is going to involve a second messenger. So you can think of the hormones as the first messenger, and then there's going to be a second messenger inside the cell that eventually leads to a change in a protein, which is going to lead to a physiological response. when the water-soluble hormone has bound to the extracellular receptor, this is going to trigger a signal transduction cascade. And cascade meaning it's going to go from one level to another, transduction meaning that it is going to transduce a signal from the extracellular fluid to the intracellular fluid. So once the hormone has bound to the receptor, it is then going to alter its conformation and alter the conformation of another protein here called the G protein, which is then going to alter the activity and shape of an enzyme called adenylate cyclase. This adenylate cyclase, just remember cyclase as in cyclic, converts ATP to cyclic AMP. So cyclase, cyclic AMP. Cyclic AMP then converts enzymes called kinases, protein kinase. Okay, it's going to activate it. So it's going to go from an inactive form to an active form. Okay, so that's what the cyclic AMP is doing. This active protein kinase now is going to take a protein, okay, and phosphorylate it. So it's now going to be a phosphorylated protein. And it's doing this by adding a phosphate group from a molecule of ATP. So ATP will be converted to ADP. The phosphate that was originally on the ATP is now on the protein. This is a common mechanism. The phosphorylation of proteins alters their function and alters the physiology of the cell. So what we've done here is we've taken an extracellular signal, okay, the hormone, and managed to alter the physiology inside the cell. Just noting that this transduction cascade amplifies the signal, because what we can appreciate here is that the binding of one hormone could produce many cyclic AMP molecules, which could activate many protein kinases and phosphorylate many proteins. So one hormone, its action is amplified by this signal transduction cascade.